Hello everyone. So today we are going to discuss cyclic photophosphorylation. So in the last session we have discussed that photosynthesis process takes place in two parts. One is light reaction and dark reaction. Light reactions takes place in two pathways. One is non-cyclic pathway and second one is cyclic pathway. Light reaction is also known as photophosphorylation. That's why these are also known as non-cyclic photophosphorylation and cyclic photophosphorylation. So we have discussed about the non-cyclic photophosphorylation, where the photolysis of water takes place, electron transfers in a non-cyclic pathway and it produces ATP, NADPH and oxygen molecule, and ATP and NADPH are used in fixing the carbon dioxide to produce the carbohydrate in dark reactions. Today we are going to discuss the cyclic pathway of photophosphorylation. So as the name suggests, it is a light induced process in which the electron travels in a cyclic pathway to produce ATP. Okay. So now we'll see the mechanism of cyclic photophosphorylation. How does it work? So what happens? Cyclic photophosphorylation involves only one photosystem, that is photosystem 1. Okay. So it involves only photosystem 1. So the spatial chlorophyll pair, the spatial chlorophyll pair of photosystem 1, that is P700. Okay, this is the spatial chlorophyll pair because it can absorb the light of wavelength 700 nanometer. So what does it do? This P700 and its LHC, that means light harvesting complexes, it absorbs the light of wavelength 700 nanometer and electron. Okay, when it absorbs this electron, this chlorophyll A molecule gets excited. That we can write as P700 star. We can denote it, the excited state, as star. So what happens when it absorbs an electron? It gets excited. That means it becomes at its higher energy level. So to return back to its original state, the original energy level, it has to transfer one electron. So it will transfer one electron to its primary electron acceptor. So here the primary electron acceptor is a modified chlorophyll. Okay, it's a modified chlorophyll and it is also the primary electron acceptor of PS1, photosystem 1. Now this electron will again transfer to a active quinone, which is denoted as A1, and this active quinone is a phylloquinone. Okay, so the electron, upon absorption of electron, it gets excited. So this electron transfers to the modified chlorophyll, and then this electron will go to this, will transfer to this phylloquinone, the active quinone of photosystem 1. This is also known as, it is phylloquinone and is also known as vitamin K1. Okay. Now the electron from this phylloquinone, it will transfer to the reaction center of photosystem 1. So what is the reaction center of photosystem 1? The iron sulfur reaction center. Okay. The reaction center is APAs or iron sulfur reaction center. It is the reaction center of photosystem 1. So the electron will transfer to the reaction center. From reaction center, again it will transfer the electron to ferredoxin. Okay. And ferredoxin is a water soluble small iron sulfur protein complexes which transfers this electron, which absorbs this electron from the reaction center. Now this ferredoxin, what happens in, we have discussed in the non-cyclic pathway that this ferredoxin transfers the electron to NADP+. Okay, NADP+, and this NADP+ reduced to NADPH. Upon conversion of the electron. NADP plus accept that electron from pyridoxine and it reduced to NADPH in the presence of FNR engine. Okay, we have this is the process, this is a part of the non-cyclic pathway. But here what happens, in cyclic pathway this NADP plus is absent. Okay, so there is no electron acceptor which can accept this electron and produce NADPH. Okay, so NADP plus is absent. Then what will happen, this pyridoxine will transfer this to plastoquinone. It will again transfer this to plastoquinone because there is no last electron acceptor, the final electron acceptor NADP is absent. So this pyridoxine will transfer this to the plastoquinone. It's again a active quinone. And then this plastoquinone will transfer this to the cytochrome B6F complex. Okay, this plastoquinone will transfer this electron to cytochrome B6F complex. As we know, cytochrome B6F complex means cytochrome B protein and cytochrome C protein. So these complex will produce, here the ADP react with inorganic phosphate and produce ATP. Adenosine diphosphate react with one inorganic phosphate and it produces adenosine triphosphate. ATP is getting produced. And after that, cytochrome B6F again transfer this to the plastocyanine. Okay, this is plastocyanine. It is also a small water soluble protein which is a blue color water soluble protein and it contains copper. Okay, and this plastocyanine will take this electron from cytochrome B6F and report it back to the photosystem 1. So now we can see that electron moves in a cyclic pathway because what happens in non-cyclic pathway from pyridoxine, NADP is present. That's why NADP accepts it and getting reduced to NADPH. It produces the final product NADPH. But here what happens, because there is no NADP, so the pyridoxine will transfer the electron to plastoquinone, then to cytochrome B6F. In cytochrome B6F, ATP is getting produced and then it will transfer the electron to plastocyanine and plastocyanine will pump the electron to the photosystem 1. Okay, to the first. So here this electron follows a cyclic pathway. That's why it is known as cyclic photophosphorylation. Okay, now we have discussed the mechanism of cyclic photophosphorylation. So the thing is, in every reaction of cyclic photophosphorylation, we get 2 ATP and 0 NADPH. That means no NADPH. Okay, we get 2 ATP. This is the product of each cyclic photophosphorylation or each cyclic reaction. Now, what is the location of this cyclic photophosphorylation? The location for this is, as we have discussed, the non-cyclic photophosphorylation takes place in the granite thylakoid, but it takes place in the stroma because in stroma there is no presence of photosystem 2. As we see, there is no involvement of photosystem 2. It only involves photosystem 1 and only photosystem 1 is available in stroma. That's why it takes place. The location is the stroma of chloroplast. Okay. So this cyclic photophosphorylation takes place in the stroma. Non-cyclic photophosphorylation takes place in the grana. So now we have discussed how this mechanism works. Then we will discuss that as we have known that non-cyclic photophosphorylation, we get ATP, NADPH and oxygen. ATP and NADPH both are required for dark reaction to fix the carbon dioxide, to produce carbohydrate, the food for the plant. And also it is a meson light reaction. So non-cyclic electron flow, non-cyclic phosphorylation actually consists of 80%, 70 to 80% of the light reaction. So it is a meson light reaction. And also in non-cyclic photophosphorylation we get 3 ATP and 2 NADPH. So we are getting more energy than this cyclic photophosphorylation, because in cyclic photophosphorylation we are getting 2 ATP. So the question is, if we are getting less ATP, less energy, and also it is not the major part of it, so still why this cyclic photophosphorylation is required, or why this photophosphorylation takes place? Okay, the question is why this photophosphorylation takes place. So now we will discuss why this photophosphorylation is required. So this cyclic photophosphorylation is predominantly found in the bacterial cells. Okay, first point, it is found in the bacterial cell, predominant in the bacteria, photosynthetic bacteria, because photosystem 2 is absent in the photosynthetic bacteria. So there is no photosystem 2 in bacteria, that's why only photosystem 1 is present, and that's why they follow this cyclic photophosphorylation. Also, plants also in plant also this cyclic photophosphorylation takes place. So, but it takes place in certain condition. In plants, it takes place in certain condition. In plants, it needs certain condition. Under certain condition only. In some condition only they follow this cyclic photophosphorylation process. So what are those conditions? First condition, if photosystem 2 is absent. Okay, first condition, if photosystem 2 is absent, then they follows this cyclic photophosphorylation. Second condition, high intensity of light. Okay, what happens in the high intensity of light, the photosystem 2 get inactivated. As we know that in photosystem 2 there are two essential membrane proteins are present, D1 protein and D2 protein. So this D1 protein get inactivated in high light intensity. Okay, and this is known as photo damage. So photo damage of photosystem 2 due to high light intensity. Okay, so the second reason is photo damage of photosystem 2 due to high light intensity, mostly the D1 protein of photosystem 2. And photosystem 2 is the main site for this photo damage. Okay, what happens? A high light intensity damage the photosystem 2. That is process is known as photo damage. PS2 or photosystem 2 is the main site of photo damage. But photosystem 1 is immune to photo damage. That's where the cyclic photophosphorylation takes place. Okay. Then the third point is, when there is ample amount of NADPH is present, but NADP is absent. The electron acceptor is absent, but NADPH is present in abundant amount. So it doesn't require more NADPH, and also NADP is not present, which can accept the electron and getting reduced to NADPH. So what happens when the NADP is not present? The electron from ferredoxin, it will go to the plastoquinone and it will follow the cyclic way. Okay, when this NADP is present, from ferredoxin, NADP accepts this electron and produce NADPH. That is the basic of non-cyclic and cyclic mechanism. So when NADP is absent, NADP absent and NADPH is more. Okay, at that point also it follows this cyclic pathway. Okay, the electron follows this cyclic pathway and the cyclic photophosphorylation takes place. Now the fourth point is low light intensity. Just like the high light intensity photodamage takes place, low light, what happens in low light intensity? Mostly in the evening time, at the low light intensity, the shutter wavelength, okay, P680, shutter wavelength P680, it does not get excited, okay, and P680 is the photosystem 2. It does not get excited. So the non-cyclic pathway starts with the excitement of photosystem 2. So when this PS2 or photosystem 2 is not getting excited, so this non-cyclic pathway cannot happen. That's why at this point also P700 get excited and this cyclic pathway takes place. Electron travels in a cyclic pathway. So no involvement of photosystem 2. Okay, the shutter wavelength does not get excited in the low light intensity and the longer wavelength get excited in the low light intensity. Okay. Now, the next point is what happens in non-cyclic photophosphorylation. In non-cyclic photophosphorylation, we know that the source of that continuous electron traveling is the photolysis of water. So, when this water getting photolyzed, it produces electron, proton and oxygen. Okay, in non-cyclic photophosphorylation, water photolyzed, it produces electron, proton and oxygen. Electron follows the non-cyclic pathway and produces ATP and NADPH. Okay, this is the non-cyclic pathway. So in this process, non-cyclic pathway, oxygen getting produced. But what happens when oxygen is getting released in more amount than the plant needed? At that point, it harms the plant in two ways. Okay, so more oxygen released in non-cyclic pathway harms the plant in two ways. One is oxidative damage of photosystem 2. First point, when more oxygen will getting released or more oxygen will get evaluated, at that time it will cause the oxidative damage to photosystem 2. And second, Rubisco enzyme. It will confuse the Rubisco enzyme. Okay. So this Rubisco enzyme, the full form, the name of this Rubisco enzyme is ribulose 1,5-bisphosphate carboxylase or oxygenase. Okay, the name is ribulose 1,5-bisphosphate carboxylase or oxygenase. Okay, this Rubisco enzyme is known as ribulose 1,5-bisphosphate carboxylase or oxygenase. This Rubisco enzyme is very essential to fix the carbon dioxide to carbohydrate. Okay, so what is the function of Rubisco? It fixes the carboxylase enzyme, so it fixes the carbon dioxide and produces carbohydrate. But also it has the capability of fixing the oxygen molecule, because as the name suggests, it is ribulose 1,5-bisphosphate carboxylase or oxygenase. It can fix the oxygen molecule. So when more oxygen is released, it will confuse the Rubisco enzyme, and this Rubisco will start fixing the oxygen molecule instead of the carbon dioxide molecule. So when the carbon dioxide molecule getting fixed, it will produce the carbohydrate. Okay, and the C3 cycle, in the Kelvin cycle under the dark reaction, the carbon dioxide produce the carbohydrate. That is the main purpose of photosynthesis process. And in that process, ATP and NADPH help it. So the main motto of it is to produce the carbohydrate. But when this Rubisco enzyme getting confused and start fixing the oxygen molecule, then carbohydrate will not be produced, and then the C3 cycle or Kelvin cycle is not taking place. That's the whole photosynthesis process is getting it is not getting any result of it. That's why this Rubisco, not to confuse this Rubisco enzyme or to avoid these circumstances, the D1 protein of the photosystem to inactivate itself. Okay, the plant inactivate the D1 protein. Plant inactivate the D1 protein of photosystem to it inactivate the D1 protein of photosystem 2. So when the photosystem 2 is inactivated and D1 protein is inactivated, that means only photosystem 1 is getting involved and the electron will travel in a cyclic path and it follows the cyclic photophosphorylation. So these are the reason why this cyclic photophosphorylation is required. Okay. And the thing is, another question is, if there is so much problem in photosystem 2, then it can also stop photophosphorylation process. But what happens? Plant need ATP and NADPH for C3 cycle or dark reaction to fix the carbon dioxide. But in other metabolic processes of the plant, ATP is also required. NADPH is present in sufficient amount, but ATP is less, because at the point when NADPH is present in sufficient amount, but plant needs more ATP for other metabolic processes. At that point, the plant activate this cyclic photophosphorylation, because in the cyclic photophosphorylation we get only ATP, no oxygen, no NADPH. Okay, so this is the reason why this cyclic photophosphorylation is required, and this is what is the mechanism of cyclic photophosphorylation. So today we have discussed the cyclic photophosphorylation mechanism and the role of the cyclic photophosphorylation, the importance of the cyclic photophosphorylation. So now we will discuss the difference between cyclic and non-cyclic photophosphorylation. So now we will discuss the difference between cyclic and non-cyclic photophosphorylation. Okay, so here we will discuss the difference between non-cyclic photophosphorylation and cyclic photophosphorylation. Non-cyclic and cyclic. Okay, as we had discussed that the non-cyclic photophosphorylation consists 70 to 80% of total light reaction. So it is the major light reaction. It is the major light reaction. And cyclic photophosphorylation takes place under certain conditions. Under certain conditions, cyclic photophosphorylation takes place, and non-cyclic photophosphorylation is the major light reaction. Then it involves both photosystems, photosystem 1 and photosystem 2. But in cyclic photophosphorylation, only photosystem 1. So it involves only photosystem 1. Okay. Next point. Here, photolysis of water takes place. Okay. In non-cyclic photophosphorylation, the source of constant electron transfer is the photolysis of water. So here, photolysis of water takes place, and here no photolysis of water, because the same electron rotate in a cyclic manner to produce ATP. So no photolysis of water molecule. In non-cyclic electron flow, the active reaction center is P680, which is the feature of photosystem 2. So in non-cyclic photophosphorylation, the active reaction center is P680, and in cyclic photophosphorylation, the active reaction center is P700. Okay, this is the active reaction center for cyclic photophosphorylation because it involves only photosystem 1. Okay, then electron travels in non-cyclic way. Okay, electron travels in a non-cyclic way, and here electron travels in a cyclic way. Okay, non-cyclic photophosphorylation, it starts with the photosystem 2. Photosystem 2 get excited, then it follows some electron acceptor to photosystem 1, and at last it produces ATP and NADPH. But here the cyclic photophosphorylation, photosystem 1 get excited, it follows some electron acceptor, and again the electron will revert back to the photosystem 1. That's why it follows a cyclic way. Here what happens? NADP acts as the electron acceptor, accept the electron and reduce to NADPH. But here no NADP involvement. In cyclic pathway, no NADP is involved, and so no production of NADPH. In this non-cyclic pathway, it produces 3 ATP. Each reaction produces 3 ATP and 2 NADPH. Okay, and here in cyclic pathway, it only produces 2 ATP. So in cyclic pathway it produces only 2 ATPs, in non-cyclic pathway it produces 3 ATPs and 2 NADPH. Then along with this ATP and NADPH, it also releases oxygen molecule. Okay, it releases oxygen molecule. But in cyclic pathway, it does not release oxygen molecule. Okay, in cyclic pathway, it does not release oxygen molecule. Now, in the last point, non-cyclic pathway is predominant. Non-cyclic pathway of light reaction is predominant in all green plants. Okay, and the cyclic pathway is predominant in photosynthetic bacteria. Okay, so these are the differences between non-cyclic photophosphorylation and cyclic photophosphorylation. So non-cyclic is a measure light reaction, whereas the cyclic photophosphorylation takes place under certain conditions. In non-cyclic photophosphorylation, both photosystems are involved, photosystem 2 and photosystem 1. In cyclic photophosphorylation, only one photosystem 1 is involved. In non-cyclic photophosphorylation, photolysis of water takes place. There is no photolysis of water. In non-cyclic photophosphorylation, the active reaction center is P680, and in cyclic photophosphorylation, the active reaction center is P700. In non-cyclic photophosphorylation, this electron travels in a non-cyclic pathway, and here it travels in a cyclic pathway. Here NADP plus accept the electron and get reduced to produce NADPH. Here NADP is absent or NADP is not involved in this process. 3 ATP and 2 NADPH is produced along with some oxygen evolution. Here only 2 ATP is produced, no NADPH. is getting produced and no oxygen molecule is getting released. It is predominant in all green plants, whereas the cyclic pathway or the cyclic photophosphorylation is predominant in all photosynthetic bacteria. So, in today's discussion we have discussed about the mechanism of cyclic photophosphorylation, why the cyclic photophosphorylation is required, and the difference between the non-cyclic and cyclic photophosphorylations. So if you have any doubt regarding today's discussion, please comment down below and don't forget to like, share and subscribe my channel. Thank you so much.